Course title: Crop Production Technology First, Kharif Crops.
Topic: Scientific Package of Practices of Paddy.
Course outcomes: arrange the basic knowledge on production technology of Kharif crops. Explain the modern technologies right from field preparation to marketing by allotting specific crop to each student. Determine the problems related to plant growth, crop production and natural resource management of Kharif crops. Analyze the different field techniques for the production of Kharif crops including sowing, irrigation, cultural practices, fertilizer and chemical application and harvesting. Evaluate the fertilizer and seed requirement. Recommended the integrated nutrient, weed and pest and diseases management programs. Design the modern techniques for growing Kharif crops in problematic areas.
Objectives of this lecture to impart knowledge about the origin, distribution, and importance of paddy. To understand the improved cultivation practices for paddy. To evaluate different varieties and cropping systems suitable for paddy. To develop skills for integrated crop management, ICM, and sustainable practices, to familiarize students with common weeds in the paddy crops and post-harvest management and marketing aspects.
Learning outcomes of this lecture. Understand the key scientific practices for optimizing paddy growth, including origin and history, soil and climate economic importance area, distribution and classification, field preparation selection of crop and variety, sowing time, seed rate and spacing method, depth of sowing, manures and fertilizer, water management, weed management, disease and pest management, harvesting and yield.
What is cereals?
Cereals were domesticated in the Neolithic, 8,000 years ago. Wheat and barley were domesticated in the Fertile Crescent. Rice was domesticated in East Asia, and sorghum and millet were domesticated in West Africa. The crop plants which belong to the family Poaceae and are grown for their edible starchy grains, just seed, called caryopsis, seed coat plus pericarp are fused or united, are called as cereals. The word cereals has been derived from Ceres' name of a Roman goddess, means giver of grains.
Now we understand the biology of cereals.
Cereals are grasses in the Poaceae family that produce edible grains. A cereal grain is botanically a caryopsis, a fruit, where the seed coat is fused with the pericarp. Grasses have stems that are hollow except at the nodes, and narrow alternate leaves borne in two ranks. The lower part of each leaf encloses the stem, forming a leaf sheath. The leaf grows from the base of the blade, an adaptation that protects the growing meristem from grazing animals. The flowers are usually hermaphroditic, with the exception of maize, and mainly anemophilous or wind-pollinated, although insects occasionally play a role. Among the best-known cereals are maize, rice, wheat, barley, sorghum, millet, oat, rye, and triticale. Some other grains are colloquially called cereals, even though they are not grasses. These pseudo-cereals include buckwheat, quinoa, and amaranth.
Scientific Package of Practices of Paddy. Scientific name Oryza sativa. Family Poaceae. Origin, Southeast Asia. Chromosome number is 24.
Economical and nutritional importance of rice. Rice is the main staple food crop for more than 60% of the world population. Around 6 to 9% protein is present in aleuron and endosperm of rice. The straw is used as cattle feed, thatching of roof and in cottage industry for preparation of hats, mats, ropes, etc. Rice husk is used as animal feed, paper making and as fuel source. Rice bran is used as cattle and poultry feed, and defatted bran, which is rich in protein, can be used in the preparation of biscuits. Rice bran oil is used in soap industry. Rice bran wax is the byproduct of rice bran oil and used in industries, no cholesterol. Nutritional value: carbohydrate 74.8%, protein 8.4%, but in milled rice 6 to 7%, fat 2.6%. Both amylase and amylopectin, more, are there. Lesser the amylase, finer the rice quality. Rice bran contains 12 to 14% protein and 20 to 25% oil.
Now we discuss about origin and classification of paddy. Origin, de Kundal, 1886, and Watt, 1862, thought that Southeast Asia was the place where cultivated rice is originated. Vavilov, 1926, suggested that India and Burma should be the origin of cultivated crop. Species, rice belongs to genus Oryza and family Poaceae. The genus includes 24 species of which O. sativa and O. glaberrima are cultivated. O. sativa has 3 subspecies viz., indica, japonica and javanica. 1. Indica, indigenous to India. It is adapted to subtropical tropical regions. In India, the varieties are very tall, photosensitive, lodging, poor fertilizer responsive, moderate filling and late maturing. The classification of paddy into 3 main groups based on species. Oryza indica, typically long-grain varieties grown in tropical and subtropical regions. Oryza japonica, short-grain varieties, common in temperate climates. Oryza javanica, medium to large-grain varieties, often grown in tropical highlands. Oryza sativa has 3 main subspecies, indica, japonica, and javanica, which can be distinguished by grain color, shape, and size. Indica is long, slender grains, e.g., basmati, mostly white when polished. Japonica is short, round grains, can be brown, unpolished, or light yellow/white, polished. Javanica is medium to large, thick grains, often less sticky and more robust in texture.
The panicle differences among the 3 subspecies of Oryza sativa are: indica, long, open, and drooping panicles with grains loosely arranged. Japonica, compact panicles with short branches, grains are closely packed. Javanica, large, open panicles with thick branches, grains are bold and bigger. Now we discuss the characteristics of Japonica and Javanica species of paddy. Japonica, it is confined to subtropical temperate regions, Japan, China, and Korea. Varieties are very dwarf, erect, non-lodging, photo-insensitive, early maturing, high-yielding and fertilizer responsive. Javanica, it is a wild form of rice and is cultivated in some parts of Indonesia. Varieties are the tallest, erect, poor filling and awned.
Now we discuss about Oryza glaberrima, commonly known as African rice, is one of the 2 domesticated rice species. It was first domesticated and grown in West Africa around 3,000 years ago. In agriculture, it has largely been replaced by higher-yielding Asian rice, O. sativa, and the number of varieties grown is declining. It still persists, making up an estimated 20% of rice grown in West Africa. It is now rarely sold in West African markets, having been replaced by Asian strains.
What is neat to understand the reasons of low yield of rice in India. Identifying production constraints, knowing whether low yield is due to poor soil fertility, water stress, pests, or diseases helps target the real problem. Improving productivity, solutions like better varieties, improved irrigation, and nutrient management can be adopted once causes are known. Efficient resource use avoids waste of water, fertilizers, and labor by using them where they are most needed. Policy planning helps government design farmer support schemes, subsidies, and research programs that address actual field issues. Food security, increasing yield ensures adequate rice supply for India's large population. Farmer income, higher productivity directly improves profitability for farmers.
Now we understand the reasons of low yield of rice in India. Widely varying climatic conditions under which rice is grown. Inefficient utilization of applied N. Limited scope for optimum water management in heavy rainfall areas. Cloudiness and its ill effects on the photosynthetic activity of rice in monsoon. Adverse effects of soil salinity or alkalinity, 0.024 m, high in AP and 7 m high in India. Susceptibility to heavy incidence of pests and diseases. Little scope for rain-fed upland rice cultivation. Indiscriminate use of fertilizers. Poor drainage, 10 lakh ha. Suffer in this zone. Monoculturing. The life cycle of the rice plant has three main stages. Vegetative stage, from germination to panicle initiation, includes seedling establishment and active tillering, with focus on leaf and root growth. Reproductive stage, from panicle initiation to flowering, includes panicle development, booting, heading, and blooming. Ripening stage, from flowering to harvest, grains fill, mature, and change color until fully ripe for harvest.
The vegetative stage of rice begins with the seedling/nursery stage, germination to transplanting, about 20 to 25 days, followed by the active vegetative stage from transplanting to maximum tillering. During the rooting stage, 4 to 10 days after transplanting, roots establish and adapt to the field. This leads to the maximum tillering stage, 35 to 42 DAT or about 60 DAS, when the plant produces the highest number of tillers. After this, in the vegetative lag stage, growth slows and the plant transitions toward the reproductive phase, marked by panicle initiation.
The reproductive phase of rice begins with the panicle initiation, PI, stage, when the growing point shifts from producing leaves to forming the panicle. This is followed by internode elongation, where the stem lengthens to support panicle emergence. In the booting stage, the developing panicle is enclosed within the flag leaf sheath, preparing for emergence. The heading stage occurs when the panicle emerges fully from the sheath, and finally, the flowering stage takes place, during which pollination and fertilization occur, determining the potential grain number.
The ripening stage of rice starts with the milk stage, 7 to 12 days after flowering, when grains are filled with a milky, fluid endosperm. It is followed by the dough stage, about 15 to 21 days after flowering, where the endosperm thickens into a soft or hard dough as starch accumulates. Finally, in the maturity stage, grains become hard, turn golden or brown, and moisture content drops to harvesting levels, marking the completion of the crop's life cycle. The life cycle of paddy begins with the vegetative stage, starting from germination in the seedling/nursery phase, 20 to 25 days, followed by transplanting, root establishment, 4 to 10 DAT, and rapid growth during the maximum tillering stage, 35 to 42 DAT, ending with the vegetative lag phase before panicle initiation. It then enters the reproductive stage, which includes panicle initiation, internode elongation, booting, heading, and flowering, where grain number is determined. Finally, the crop moves to the ripening stage, progressing from the milk stage, 7 to 12 DAF, to the dough stage, 15 to 21 DAF, and ending at maturity, when grains harden and are ready for harvest.
Area and distribution. Rice is cultivated across tropical, subtropical, and warm temperate regions, extending from 40 degrees south to 50 degrees north latitude. The majority of rice-growing areas are situated between the equator and 70° 140° longitude. The highest yields are recorded between 30 degrees and 45 degrees north latitude, while in India, China, and Egypt, located between 21 degrees and 30 degrees north latitude, average yields range from 2.0 to 5.7 t/ha. In contrast, countries closer to the equator typically record lower yields of 0.8 to 1.4 t/ha. In India, rice occupies about 24% of the gross cropped area, highlighting its importance in the nation's agriculture. Rice accounts for 42% of the total food grain production and 45% of the total cereal production in India. Over the past 6 decades, its production has increased by about 3.5 times, rising from 250.3 lakh tons during the first 5-year plan to 857.3 lakh tons in the 10th plan period. While the productivity of rice in India is higher than that of Thailand and Pakistan, it remains significantly lower than that of Japan, China, Vietnam, and Indonesia. India's share in global rice production has consistently ranged between 19.5% and 24.52%.
India has the largest cultivation area, 44 to 45 million hectares, but relatively low productivity, 2.5 to 3.0 tons per hectare. China grows rice on 30 to 32 million hectares but has high productivity, 6.5 to 7.0 tons per hectare, producing 200 to 205 million metric tons. Global total rice production is 738 to 745 million metric tons from 160 to 165 million hectares, with average productivity of 4.5 to 5.0 tons per hectare. Indonesia, Thailand, and Vietnam have moderate productivity, 3.5 to 5.5 tons per hectare. Bangladesh has smaller area and lower productivity, 3.0 to 3.5 tons per hectare. United States has the smallest area but the highest productivity, 7.5 to 8.0 tons per hectare.
Climatic requirement of paddy. Rice can be cultivated in a wide range of locations and climates. Indica varieties are predominantly grown in tropical regions. The crop thrives in hot, humid conditions and is best suited to areas with high humidity, prolonged sunshine, and a reliable water supply. Key climatic factors such as temperature, solar radiation, and rainfall significantly influence rice yield, both directly, by affecting the plant's physiological processes involved in grain formation, and indirectly, by impacting the incidence of pests and diseases.
Rice requires specific temperature conditions for optimal growth, with critical lower and upper limits generally below 20 degrees Celsius and above 30 degrees Celsius, respectively. Germination improves linearly as temperatures rise within the 22 to 31 degrees Celsius range. Just after germination, temperature influences growth rate, while later it slightly affects tillering and relative growth. During the reproductive stage, cooler temperatures increase the number of spikelets per plant. Overall, temperature impacts grain yield by affecting tillering, spikelet formation, and ripening.
The cardinal temperature requirements for rice at different growth stages, including the low, high, and optimum temperature ranges for each stage. Germination occurs between 16 to 19 degrees Celsius, low, and up to 45 degrees Celsius, high, with optimum 18 to 40 degrees Celsius. Seedling emergence and establishment need 12 to 15 degrees Celsius minimum, 35 degrees Celsius maximum, and 25 to 30 degrees Celsius optimum. Rooting thrives best at 25 to 28 degrees Celsius, with limits of 16 degrees Celsius, low, and 35 degrees Celsius, high. Leaf elongation requires 7 to 12 degrees Celsius minimum, 45 degrees Celsius maximum, and about 31 degrees Celsius optimum. Tillering happens between 9 to 16 degrees Celsius and 33 degrees Celsius, with 25 to 31 degrees Celsius as optimum. Panicle initiation needs temperatures above 15 degrees Celsius, but no exact high or optimum is given. Initial flowering prefers 18 to 28 degrees Celsius, with limits of 15 to 20 degrees Celsius, low, and 30 degrees Celsius, high. Anthesis, flower opening, has an optimum of 30 to 33 degrees Celsius, tolerating 22 degrees Celsius low and 36 degrees Celsius high. Ripening requires 20 to 29 degrees Celsius optimum, with limits of 12 to 18 degrees Celsius low and 30 degrees Celsius high.
Soil requirement. Rice can grow in almost any soil type, but its requirement for standing water during much of its growth makes heavy soils, like clay and clay loam, more suitable because they hold water well. Although rice can survive in both acidic and alkaline soils, it produces higher yields in soils that are slightly acidic, pH 5.5 to 6.5. Sandy soils can also be used if water is adequately supplied and retained, but they are less ideal compared to heavier types.
Rice growing seasons. Aus or Ashu, pre-monsoon crop. Meaning, Ashu means quick. Sowing, pre-monsoon period, around March-April. Harvest, August-September. Feature, short duration crop, suited to early rains and areas with limited irrigation. Aman or Kharif, monsoon winter crop. Meaning, refers to safety or stability. Sowing, June, beginning of monsoon. Harvest, winter season, November-December, sometimes January. Feature, this is the main rice crop in many regions, benefiting from abundant monsoon rains. Boro or summer crop. Sowing, January-February. Harvest, April-May. Varieties, Gautam, Krishna, Hasma. Feature, grown mainly with irrigation during the dry season, high yielding if water supply is assured.
Genetic improvement in paddy. T.N.1. Cross, DG Wu Gen, dwarf, nitrogen responsive, times to Sai Young Chung, tall, drought resistant. IR 8. Cross, DG Wu Gen times PETA, brown tipped short legend. Released, 1966. Yield, 60 to 80 Q/Ha. Duration, 140 to 145 days. Jaya. Cross, TN, 1 times T141. Jagannath. Developed as a mutant of T141. Pusa basmati 1. World's first scented high-yielding variety from IARI. Pusa RH10. First fine scented rice variety. PHB 71. First hybrid scented rice variety.
Hybrid rice. First hybrid rice was developed using cytoplasmic male sterility, CMS, restore gene in China. In India, hybrid rice development began in 1994. Examples by region. Andhra Pradesh, APRH1, APRH2. Karnataka, KARH1. Tamil Nadu, MGR1. Bihar P6444, PA6201, DRH1. Golden rice. Contains beta-carotene, provitamin A. Used to treat xerophthalmia, partial or complete blindness caused by vitamin A deficiency. Developed by Professor Ingo Patricus, Zurich, and Professor Peter Bayer, Germany. Genes used. From daffodil, Narcissus pseudonarcissus, and Bacterium Erwinia urdivora, encode beta-carotene. Agrobacterium tumefaciens, crown gall bacterium, provided plasmids as gene carriers into rice tissue.
Rice ecosystems in India are classified based on land type and water management into lowland, wetland, and upland, dryland systems, with three principal cultivation methods: dry system, upland, where rice is direct seeded in unbunded, rain-fed fields without standing water; semi-dry system, where fields are prepared under dry conditions, rice is direct seeded or transplanted, and irrigation or rainfall later creates saturated soil; and wet system, lowland, where burnt fields are puddled and rice is grown under continuous standing water from monsoon rains or irrigation, ensuring higher yields but requiring more water.
Now we discuss about the rice ecosystem's dry system, or upland rice, is mainly cultivated in the eastern states of India, such as Assam, West Bengal, Odisha, Bihar, and Uttar Pradesh, as well as parts of central India, including Madhya Pradesh, portions of Andhra Pradesh, and Maharashtra. This system involves growing rice in unbunded, rain-fed fields without prolonged standing water, making it dependent on early monsoon rains. Locally, it is known by different names: Aus in West Bengal, Aus or Ahu in Assam, Biali in Odisha, and Paddy or Kwari in Uttar Pradesh, and is typically sown before or at the onset of monsoon, using early maturing, drought-tolerant varieties.
Field preparation. The field is ploughed and harrowed to achieve fine tilth, leveraging the summer rains and early monsoon showers. Gypsum is applied at 1 tonne per hectare, and FYM or compost is incorporated at 12.5 tonnes per hectare. Varieties: Turanta, Prabhat, Saket 4, Saroj Dan Lakshmi, Sushk Smrat, Siardan, 40, Abhishek. Seed rate and seed treatment. The seed rate is 75 kilograms per hectare. Seeds are treated with a fungicide, Bavastine or Therum, at 2 grams per kilogram 24 hours before sowing. Additionally, azospirillum is applied at 3 pockets, 600 grams per hectare of seed. Season, May to June.
In upland paddy cultivation, several sowing methods are used depending on resources and field conditions. Broadcasting involves scattering seeds evenly over a well-prepared seedbed, requiring later thinning for uniform spacing. Dibbling places seeds directly into small holes at regular intervals, ensuring better plant spacing and seed-soil contact. Sowing behind the country plow or using a seed drill allows seeds to be dropped into shallow furrows, often in continuous lines. Line sowing, done at about 20 cm row spacing, facilitates easier weeding and better aeration. After sowing, thinning and gap filling should be carried out around 10 to 12 days after sowing, DAS, to maintain the recommended plant population and ensure even crop growth.
For nutrient management in upland paddy, the recommended fertilizer dose is 80 kg nitrogen, 40 kg P2O5 and 40 kg K2O per hectare. The full dose of phosphorus, P, and potassium, K, along with half of the nitrogen, N, should be applied at the time of the last plowing to ensure good early growth. The remaining nitrogen, 20 kg per hectare each, is applied in 2 equal splits, the first at 20 to 25 days after sowing, DAS, to support tillering, and the second at 40 to 45 DAS to promote panicle initiation and grain development. This split application ensures efficient nutrient use, minimizes losses, and matches the crop's growth requirements.
Weed management in dry or upland rice. Under upland conditions, weeds can reduce yield by up to 50%. To manage weeds effectively. First weeding, perform the first weeding at 15 to 20 days after sowing, DAS. Second weeding, the second weeding should be done around 45 DAS. Herbicide application. Apply thiobencarb at 2.5 L/Ha or pendimethalin at 3.0 L/Ha as a sand mix on the 8th DAS. Additionally, apply bispirabac sodium at 40 g active ingredient, AI, or Nomini Gold, 400 ml per hectare, mixed with 600 liters of water/Ha at 18 to 20 DAS.
Semi-dry rice ecosystem. This system is practiced in areas where the crop initially grows under upland, dry conditions but later transitions to wetland conditions. Rice is first sown and managed like dry upland rice, relying on early monsoon showers. After 45 to 60 days after sowing, thus, when heavy rains arrive and fields become waterlogged, the crop is plowed crisscross to control weeds and adjust plant spacing. This process is called bushing, which helps maintain proper plant population and reduce competition from weeds. After bushing, the crop is managed using wetland, lowland, rice practices until maturity. Season, May-June sowing. Recommended varieties, 90 to 110 days duration Turanta, Prabhat, Saket 4, Saroj, Dhanlakshmi, Sushmrat, CR Dhan 40, Abhishek, Sita, Kanak, IR 36, Rajendra Sweta, Sahbhagi. Seed rate, 75 kg per hectare. Seed treatment. Treat seeds with bavistine or thiram at 2 g per kg of seed, 24 hours before sowing, to protect against seed-borne diseases. Inoculate seeds with azospirillum at 3 packets, 600 g, per hectare seed rate to improve root growth, nitrogen fixation, and early vigor. Sowing methods. Broadcasting, scattering seeds uniformly across the field, simplest method but may lead to uneven spacing. Line sowing, sowing seeds in rows at 20 cm spacing, preferred over broadcasting as it ensures better plant spacing, easier weeding, and higher yield. Dibbling, placing seeds directly in holes at fixed intervals, ensuring accurate spacing. Sowing behind the country plow, dropping seeds into shallow furrows made by the plow, often followed by light covering of soil.
After cultivation in semi-dry rice. After sowing, thinning and gap filling should be carried out 10 to 12 days after sowing, DAS, preferably soon after a rainfall when the soil is moist, to ensure uniform plant population and healthy crop establishment. Fertilizer management. Recommended dose, 80 kg nitrogen, 40 kg P2O5 and 40 kg K2O per hectare. Application method. Apply the full dose of phosphorus, P, and potassium, K, along with half the nitrogen, N, at the time of last plowing to support early seedling growth. The remaining nitrogen should be applied in 2 equal splits. 20 kg N per ha at 20 to 25 DAS to promote tillering. 20 kg N per ha at 40 to 45 DAS to support panicle initiation and grain formation.
Weed management in paddy cultivation. 1. Cultural practices. Timely weeding. Regular and timely weeding is essential to control weed growth before they become competitive with the crop for nutrients, water, and light. Soil preparation. Proper soil preparation, including plowing and harrowing, helps in creating a fine tilth and reduces weed seed bank in the soil, making it easier for seedlings to establish. Planting methods. Using methods like dayporg nursery or direct sowing can reduce weed infestations by promoting early and uniform seedling establishment. A well-established crop canopy can also outcompete weeds. Crop rotation. Rotating rice with other crops, such as legumes, can break the weed cycle, reduce weed pressure, and improve soil health. This reduces the likelihood of weeds becoming resistant to herbicides. Soil moisture management. Maintaining optimal soil moisture is crucial for weed control. For paddy, standing water after sowing can suppress certain weed species that do not thrive in flooded conditions. Regular water management prevents weeds from thriving. Water management. Proper water management, such as maintaining 2.5 cm of standing water after the initial saturation period, suppresses weed germination, particularly for weed species that are sensitive to waterlogging. 2. Mechanical control. Hoeing and hand weeding. These are labor-intensive but effective methods for weed control, especially in small or irregularly shaped fields. Hand weeding can be done between rows to manually remove weeds without damaging the rice seedlings. Rotary weeding. This mechanical method involves using a rotary weeder to control weeds in the field. It's effective for controlling shallow-rooted weeds in the early stages of crop growth. However, it should be done carefully to avoid disturbing the rice roots. 3. Herbicide application. Thiobencarb or pendimethalin application. Apply thiobencarb at 2.5 litre per ha or pendimethalin at 3.0 litre per hectare as a sand mix at 8 DAS if adequate moisture is available. This helps to control weeds early in the crop's growth stage. The application should be followed by one-hand weeding at 30 to 35 DAS to manage any remaining weeds and ensure the crop remains competitive. Bispiribac sodium. Apply bispiribac sodium at 40 gram active ingredient per ha or Nomini Gold at 400 milliliters per hectare when weed pressure is high. Healthy growth and prevent stress.
Wet or lowland rice system puddling. In the wet or lowland rice system, fields are kept flooded for most of the crop's life cycle, and puddling is a key land preparation practice. Puddling refers to the mechanical manipulation of soil under high moisture conditions to create a soft, muddy layer that minimizes water loss by reducing deep percolation. This is achieved by churning and compacting the soil while it is saturated. Objectives of puddling: reduce percolation losses by forming a compact subsoil layer that holds water. Control weeds by burying them and making conditions unfavorable for germination. Facilitate transplanting by creating a soft bed for easy seedling placement. Improve nutrient availability by reducing leaching losses. Enhance root establishment through better soil-water contact. Implements used: puddling can be performed using animal-drawn plows, power tillers, or tractor-drawn implements, depending on equipment availability and soil type.
राइस में जो पडलिंग है वो की जा रही है। इससे क्या होता है कि जो मिट्टी और जो पानी होता है उसको अच्छे से मिक्स कर दिया जाता है इस पडलिंग प्रोसेस के द्वारा, जिससे कि पोर स्पेस वगैरह बंद हो जाते हैं और राइस की फील्ड में पानी आराम से भरा होता है। तो आप ये देख रहे हो टायर के ऊपर ये जो व्हील गेज लगा हुआ है, इससे इसको मिक्स किया जाता है।
Preparation of nursery for sowing, wet rice. Wet rice cultivation requires a well-puddled nursery bed to ensure healthy seedlings for transplanting. Main field preparation for transplanted rice. The land should be plowed repeatedly 3 to 4 times, with an interval of about 4 days between each puddling. This allows proper soil softening and decomposition of organic matter. Implements used: animal-drawn, country plow or mold-board plow. Mechanized, tractor-drawn cage wheel or power tiller. During puddling, a standing water depth of 3 to 5 centimeters should be maintained to keep the soil saturated, making it easier to work the field and reducing weed growth. Optimum puddling depth, about 10 centimeters for clay and clay loam soils, deep enough to break clods, form a compact layer to reduce percolation, and create a soft surface for transplanting.
The dry, wet, and daypog methods, along with modified mat and bubble tray nurseries, are techniques used in paddy cultivation to improve seedling growth and establishment. In the dry method, seeds are broadcasted on dry soil and irrigated later, whereas in the wet method, seeds are sown in standing water, promoting faster germination. The daypog method involves sowing seeds on raised nursery beds, ensuring good aeration and preventing waterlogging, which helps produce strong seedlings. The modified mat nursery method involves growing seedlings on mats made of jute or synthetic materials, ensuring uniform growth and easy transplantation. The bubble tray nursery is a more recent technique, where seedlings are grown in trays with small pockets and oxygenated water, ensuring high germination rates and healthy roots. These methods, by enhancing seedling quality and reducing the risk of diseases, ultimately result in improved transplanting success and better paddy yield.
Dry and wet nursery method. For both the dry and wet nursery methods, 5 to 8 meter long, 1.2 meter wide, and 0.30 meter raised beds are prepared. In the dry method, seeds are sown on the raised beds, and frequent irrigation is applied to promote seed germination. Fertilizer is applied at the rate of 1 kg nitrogen, 0.50 kg P2O5 and 0.50 kg K2O per 100 square meters, ensuring balanced nutrient supply for healthy seedling growth. For the wet method, the process is similar, but the soil is kept waterlogged for seed germination. The beds remain flooded to facilitate seedling growth under wet conditions. Daypog nursery method. In the daypog method, raised beds of the same dimensions, 5 to 8 meter long, 1.2 meter wide, and 0.30 meter high, are prepared, and seeds are broadcast at a rate of 1 kg per m superscript 2. This requires about 40 to 45 square meters of nursery area for raising seedlings for 1 hectare of paddy. Fertilizer is applied at a rate of 2.5 kilograms nitrogen, 1.50 kilograms P2O5 and 1.50 kilograms K2O per 45 square meters to provide the necessary nutrients. After about 12 to 14 days, when the seedlings reach the 2 to 3 leaf stage, they should be pulled out carefully and transplanted into the main field.
Seed rate for different rice varieties. For optimal growth, the recommended seed rates are as follows: 30 kilograms per hectare for fine rice varieties, 40 kilograms per hectare for medium rice varieties, 50 kilograms per hectare for coarse rice varieties. Pre-treatment of seeds before sowing. To protect seeds from fungal infections and enhance germination, treat the seeds with a fungicide at 2 g per kg of seed. Suitable fungicides include thiram, captain, carboxin, or carbendazim. The seeds should be treated at least 24 hours before soaking for sprouting. After treatment, the seeds can be stored for up to 30 days without significant loss in viability.
Seed treatment for paddy cultivation. Wet seed treatment. Treat the seeds in a solution of carbendazim, pyroquinone, or tricyclazole at a concentration of 2 gram per liter of water for every 1 kilogram of seed. Soak the seeds in this solution for a specified time to protect against fungal diseases and improve germination. Azospirillum seed treatment. For enhanced nitrogen fixation, mix 3 packets, 600 g ha of azospirillum culture with enough water to soak the seeds overnight before sowing them in the nursery bed. This promotes healthy root development and strengthens the plant's growth. Pseudomonas fluorescens seed treatment. Similarly, mix 3 packets, 600 gram per ha of Pseudomonas fluorescens culture in water. Soak the seeds overnight before sowing in the nursery bed. This treatment helps control seed-borne diseases and boosts plant health by promoting beneficial microorganisms in the soil.
Water management in nursery. For proper water management in the nursery, begin by draining the water 18 to 24 hours after sowing to allow the seeds to settle. From day 3 to day 5, ensure the soil is adequately saturated with water to facilitate seedling establishment. Starting from day 5, gradually increase the water level to a depth of 1.5 centimeters, adjusting based on the height of the seedlings. Once the seedlings are established, maintain a consistent water depth of 2.5 centimeters to promote healthy growth and prevent stress.
In this video, we are going to see about mat nursery preparation of SRE technique of rice practiced in MIT College of Agriculture and Technology form. As per this technique, we need nursery area up to a requirement. Before preparing the mat nursery, apply farmyard manure and puddle the land with the help of puddler and level the land. After leveling, the field should be flooded. Materials required: farmyard manure, vermicompost, azospirillum, paddy straw, banana leaf, polythene sheets, paddy seeds, and wooden frames. Here, we are using a long duration rice variety BPT 5204. Prior to sowing, soak the seeds overnight with water in gunny bags and treat the seeds with biofertilizer or biocontrol agents. Let it be free for germination. Drain the excess water before raising the bed of 1 feet height, 1.25 meter width and convenient length. Spread banana leaf on nursery bed to prevent the root penetration into a soil. Sometimes, plastic sheets can also be used altered for banana leaf to prevent the root penetration. Laying of soil mixture is very important to nursery. Spread alternate layers of soil and vermicompost over the polythene sheet or banana leaf. Spread the mixture evenly on the bed. Place the wooden frame and fill it with a mixture of soil and vermicompost and level it. Broadcast the pre-germinated seeds on the bed. Sprinkle the seed vermicompost over the seeds. Cover it with a paddy straw as a mulch. Finally, watering it with a rose can for germination. After germination, transplant the seedlings at 2-leaf stage. 8 to 12 days of young seedlings. Square planting and single seedling per hill is mandatory. Practice alternate wetting and drying of irrigation for the better growth. After 45th day of transplanting, here we can see the beautiful view of this rice field. The seedlings are now grown up to a late vegetative stage or rooting stage. Irrigate the field at 2.5 cm under level of stagnation condition. Every week interval, the tillers are to be counted with length and breadth of leaf, and leaf area index is also measured to estimate the crop growth. First weeding is done at 25th day after transplanting. Followed by bund clearing is also to be done. This will reduce the competition between feed and crop and enhance the yield, which leads to increase in productivity. After clearing the bunds and field from weeds, they are collected and destroyed to avoid the pest and disease infestation in the rice field. After weeding, apply second dose of fertilizer as urea complex, which is mixed with neem cake at correct proportion. For nutrient management of SRI, 150 kg of urea, 50 kg of SSP and 50 kg of MOP is required.
Weed management in paddy. Weed management in paddy is essential for maintaining high yields, as weeds compete with rice plants for nutrients, sunlight, and water. An integrated approach combining cultural, mechanical, and chemical methods ensures effective control. 1. Cultural practices. These practices aim to prevent or minimize weed growth by creating unfavorable conditions for weeds. Timely weeding. Remove weeds during the early growth stages of paddy to prevent competition when rice plants are most vulnerable. The first weeding is usually done around 20 to 25 days after sowing or transplanting, followed by subsequent weeding as required. Soil preparation. Proper land preparation through plowing, puddling, and leveling helps bury weed seeds and disrupt their germination. Puddling in wetland rice also reduces weed growth by limiting oxygen availability to weed seeds. Planting methods. Use methods like line transplanting, drum seeding, or mat-type nursery transplanting to facilitate easier weeding and uniform plant growth. Crop rotation. Rotating rice with non-rice crops, such as pulses or oilseeds, helps break weed life cycles and reduces the buildup of specific weed species. Soil moisture management. Maintain optimal soil moisture to favor rice growth and suppress certain weed species. Water management. Keep fields flooded, 3 to 5 centimeters water depth, during early crop stages to suppress most upland weed species. Avoid frequent drying, which promotes weed emergence. 2. Mechanical control. These methods physically remove or damage weeds. Hoeing and hand weeding. Manual removal of weeds using a hoe or by hand, especially effective in small fields and where labor is available. Rotary weeding. Use of rotary weeders between rows in line-planted fields. This method also aerates the soil and improves root growth. 3. Chemical control. Herbicides can effectively control weeds when used properly. Pre-emergence herbicides. Apply any one of the following herbicides 8 days after sowing, DAS, in the nursery to control weed germination. Butacla, 2.0 liters per hectare. Thiobencob, 2.0 liters per hectare. Pendimethalin, 2.5 liters per hectare. Anilafos, 1.25 liters per hectare. Ensure uniform application and maintain a thin water film, 2 to 3 cm, in the field during application.
Application of fertilizer and micronutrients in lowland paddy. Recommended fertilizer dose. Short duration varieties, 100 kg nitrogen, 40 kg P2O5 and 30 kg K2O per hectare. Medium to long duration varieties, 120 kg nitrogen, 60 kg P2O5 and 40 kg K2O per hectare. Phosphorus, PO, and potassium, KO. Application: apply the entire recommended dose of PO and KO as basal during puddling. Exception: coarse textured soils, low CEC. Apply KO in 2 equal splits, 50% at basal and 50% at maximum tillering or panicle initiation stage. Nitrogen N application in clay soils. Apply in 3 splits, 50% basal plus 25% at maximum tillering plus 25% at panicle initiation stage. Micronutrient management, zinc. For zinc deficiency, dip rice seedlings roots in 1% ZNSO solution before transplanting. Alternatively, apply ZNSO at the rate of 25 kg per ha to the field. Application of organic manures in paddy. Farmyard manure, FYM, or compost. Apply 10 tons per ha of well-decomposed FYM or compost. Spread the manure uniformly on dry soil before applying irrigation water to the field. Incorporate it into the soil during field preparation for better nutrient availability. If FYM or compost is not available, apply green manure or green leaf manure at 6.25 tons per ha. These improve soil fertility by adding organic matter and nitrogen. If green manuring is not possible, apply vermicompost at 4 tons per ha, which provides readily available nutrients and beneficial microbes.
Incorporation of green manure in paddy. Selection and cultivation of green manure. Stem nodulating Sesbania rostrata is ideal for paddy fields and can be sown during April-May. Use a seed rate of 50 to 60 kg per ha. Grow for 45 to 50 days before incorporation to maximize biomass and nitrogen fixation. Incorporation into the soil. Plow or incorporate the green manure crop, or green leaf manure, directly into the soil using a moldboard plow or tractor. Incorporation should be done while the crop is green and succulent for better decomposition. Phosphorus management. When green manure is used, rock phosphate can be applied as a cost-effective source of phosphorus since decomposition improves P availability. Seedling density and planting geometry. Wet nursery, 3 to 4 seedlings per hill. Dayporg method, 6 to 8 seedlings per hill. Saline soils, 4 to 6 seedlings per hill. Plant density and spacing may vary depending on soil fertility, genotype, and local conditions.
Application of biofertilizers in paddy. Azolla. Azolla is a free-floating water fern that harbors anabina, a nitrogen-fixing cyanobacterium, in its leaves. It enriches the field with nitrogen, reduces chemical N fertilizer use, and also provides organic matter. Blue-green algae, BGA. Apply powdered BGA at 10 kg per hectare by broadcasting evenly over the field. This enhances nitrogen availability through biological fixation. Root dipping in azospirillum slurry. Prepare a slurry using 5 pockets, total 1000 gram per hectare, of azospirillum inoculant mixed with 40 liters of water. Dip the roots of rice seedlings in this slurry for 15 to 30 minutes before transplanting. This promotes early root establishment and enhances nutrient uptake. Soil application of azospirillum. Mix 10 pockets, total 2000 gram per hectare, of azospirillum inoculant with 25 kg of FYM and 25 kg of soil. Broadcast the mixture uniformly in the main field before transplanting. Electric light bulb benefits of biofertilizers. Reduce dependency on chemical nitrogen fertilizers. Improve soil microbial diversity and activity. Enhance plant vigor and yield sustainably.
Water management in paddy fields. Total water requirement. Rice requires about 1,800 to 2,000 millimeters of water during its growth cycle. Water depth at different stages. At transplanting, maintain a shallow water depth of 2 cm to reduce transplanting shock. First 7 days after transplanting, continue maintaining 2 cm depth to aid seedling establishment. Establishment to maturity, maintain 5 cm submergence consistently throughout crop growth, except during specific management practices like fertilizer application or weeding, when water can be temporarily drained. Critical stages for water requirement. Adequate water must be ensured at the following stages to avoid yield loss. Primordial initiation, start of panicle development. Booting stage, swelling of the flag leaf sheath. Heading stage, emergence of panicle. Flowering stage, anthesis. Milk stage, grain filling with milky fluid.
Now we discuss the nitrogen losses in paddy. The different pathways through which nitrogen, N, can be lost in agricultural systems. Nitrogen is an essential nutrient for plant growth, but it is not fully utilized by crops due to various physical, chemical, and biological processes. The figure highlights these loss mechanisms. Denitrification loss. Microorganisms in anaerobic, low oxygen, soil conditions convert nitrate, NO, into gaseous forms like nitrogen gas, N, and nitrous oxide, NO, which escape into the atmosphere. Common in waterlogged or compacted soils. Fixation by microbes. Soil microbes can immobilize available nitrogen by incorporating it into their biomass, temporarily making it unavailable to plants. Runoff and leaching loss. Runoff, surface water flow removes nitrogen from fields, especially during heavy rains or over-irrigation. Leaching, nitrate moves down through the soil profile beyond the root zone with percolating water, often contaminating groundwater. Volatilization loss. Loss of nitrogen as ammonia gas, NH, into the atmosphere, especially when urea is surface applied without incorporation into the soil. Crop uptake. Plants absorb nitrogen for growth, but this is not a loss in the negative sense, rather, it's the intended pathway. However, any nitrogen taken up by weeds or non-target plants can be considered a diversion from the intended crop. Ammonium fixation. Ammonium, NH, ions get trapped between clay mineral layers, making them temporarily unavailable to plants. Reducing nitrogen losses in rice cultivation. Efficient nitrogen, N, management is essential to improve nitrogen use efficiency, NUE, and minimize environmental losses. The following strategies are commonly adopted. Split application of nitrogen. Applying N fertilizer in 3 to 4 split doses according to soil type helps synchronize N availability with crop demand, thereby enhancing NUE. Use of slow-release fertilizers. Slow-release forms of urea reduce rapid N losses and provide a steady nutrient supply. Examples include IBDU, isobutylidene diurea, UF, urea formaldehyde, sulfur-coated urea, neem-coated urea, gypsum-coated urea, mud ball urea. Deep placement of urea supergranules. Larger urea granules are placed below 10 cm m depth in the reduced zone of the soil, limiting volatilization and denitrification losses. Use of nitrification inhibitors. Compounds such as AM and NSERV suppress the activity of nitrosomonas and nitrobacter, slowing the conversion of ammonium, NH, to nitrate, NO, thereby reducing leaching and denitrification. However, these inhibitors are currently not commercially available in India. Effective weed control is essential in paddy cultivation to ensure healthy crop growth and higher yields. A combination of cultural, mechanical, and chemical methods is often used. Summer plowing and puddling. Deep plowing during summer exposes weed seeds and rhizomes to intense sunlight, reducing their viability. Puddling, wet tillage, before transplanting creates anaerobic conditions that suppress weed germination and growth. Straight row planting. Planting paddy in straight rows facilitates easier identification and removal of weeds through mechanical or manual methods. It also improves the efficiency of herbicide application. Flooding at effective root depth. Maintaining water levels at the effective root depth creates unfavorable conditions for most upland weeds. Continuous or intermittent flooding reduces germination of many weed species. Manual weeding. Hand pulling or weeding is an effective method for small plots. However, it is labor-intensive, time-consuming, and less economical for large-scale cultivation. Chemical weed control. Herbicide application is a quicker and less laborious option, allowing large areas to be treated in a short period with limited labor. It is often more cost-effective in terms of labor savings. Limitations. No single herbicide is effective against all weed species. The initial investment cost is relatively high. Risk of herbicide resistance and potential environmental concerns require careful selection and use. Integrated weed management, IWM, combining cultural practices like puddling and flooding, mechanical methods like cono weeding, and selective use of herbicides offers the most sustainable and effective approach.
Integrated weed management, IWM, in paddy. Integrated weed management in paddy combines chemical control with cultural and mechanical practices to achieve effective and sustainable weed suppression. Pre-emergence herbicide application, 3 days after transplanting, DAT. Apply one of the following herbicides uniformly over the field. Butaclar at 2.5 liters per hectare. Thiobencob at 2.5 liters per hectare. Pendimethalin at 3.0 liters per hectare. Anilafos at 1.25 liters per hectare. These herbicides prevent the emergence of weed seedlings by inhibiting germination or early growth. Pre-emergence herbicide mixtures. To broaden the weed control spectrum, use mixtures of a pre-emergence herbicide with a broadleaf weed killer. Butaclar 1.20 liters per ha plus 2 for DEC 1.5 liters per hectare. Thiobencob 1.20 liters per ha plus 2 for DEC 1.5 liters per hectare. Pendimethalin 1.5 liters per ha plus 2 for DEC 1.5 liters per hectare. Mix the herbicides with sand for uniform distribution in the field. Water management during application. Maintain a water depth of 2.5 cm at the time of herbicide application. Avoid draining water for at least 2 days after spraying. Do not provide fresh irrigation during this period to ensure maximum herbicide efficacy. Post-emergence herbicide option. If pre-emergence herbicides are not applied, control weeds with a post-emergent spray. 2 for D-sodium salt, fernaxone 80% WP, at 1.25 kg per hectare. Dissolve in 625 liters of water and spray uniformly 3 weeks after transplanting using a high-volume sprayer.
Harvesting paddy at the optimum time is crucial in tropical regions to avoid yield and quality losses. Delayed harvesting can lead to grain shedding, scattering, lodging, bird damage, and deterioration in grain quality due to overmaturity. Timely harvesting ensures high grain quality with better milling recovery, higher market value, improved consumer acceptance due to uniform grain appearance and cooking quality. In India, studies have shown that harvesting between 27 and 39 days after flowering results in maximum head rice recovery. The ideal grain moisture content at harvest is 18 to 20%. Pre-harvest preparations. Drain excess water from the field 7 to 10 days before the expected harvest to facilitate easier field operations and minimize grain sprouting. Monitor crop maturity indicators. Around 80% of panicles should have turned straw yellow. Most grains at the base of the panicle in selected tillers should be in the hard dough stage. Special practice for early maturity. Spraying 20% sodium chloride (NaCl) solution 1 week before harvest can hasten maturity by 3 to 4 days, helping the crop escape damage from untimely monsoon rains.
Post-harvest technology of paddy. Post-harvest operations in paddy aim to minimize quantitative and qualitative losses, maintain grain quality, and enhance market value. Proper handling after harvest is essential to retain milling yield, cooking quality, and storability. Threshing. The process of separating grains from the harvested panicles. Traditionally done by beating the panicles against a hard surface or treading by animals, modern methods involve mechanical threshers, which are faster, labor-saving, and reduce grain breakage. Cleaning. Removal of impurities such as straw, chaff, stones, weed seeds, and dust to improve grain quality. Achieved through winnowing, manual or mechanical, and sieving systems. Grading. Sorting of grains based on size, shape, and quality to ensure uniformity for milling and marketing. Grading enhances consumer acceptance and helps meet export standards. Drying. Reduces grain moisture to a safe storage level, 12 to 14%. To prevent fungal growth, insect attack, and sprouting. Sun drying is common in rural areas. Mechanical dryers are used for large-scale operations, ensuring uniform drying and better quality. Parboiling. A hydrothermal process involving soaking, steaming, and drying of paddy before milling. Improves milling recovery, hardens kernels to reduce breakage, enhances nutritional value, and increases storage life. Curing and preservation. Curing refers to the aging of rice to develop better cooking qualities and aroma. Preservation includes fumigation, use of hermetic storage, and periodic inspection to control pests and maintain grain quality during storage. Processing. Involves milling, removal of husk and bran layers, to produce brown rice, white rice, or other rice-based products. Modern mills ensure high recovery of head rice with minimal broken grains. Packing. Proper packaging protects rice from moisture, pests, and contamination. Common materials include jute bags, HDPE/PP woven sacks, and hermetic storage bags for long-term preservation. Transportation and marketing. Grains should be transported under dry, clean conditions to avoid spoilage. Efficient marketing channels connect producers to wholesalers, processors, and retailers, ensuring fair pricing and reduced post-harvest losses.
Now we discuss about the rice processing sequence from harvesting to final products. Spikelet, harvesting stage. The rice plant produces spikelets that contain the grain. At harvest, the spikelets are cut and collected. The pedicel, stalk portion, is separated from the spikelet during threshing. Paddy rice. After threshing, the grain with its husk intact is called paddy rice. This is the raw form of harvested rice. Dehusking. The husk, outer protective layer, is removed mechanically or traditionally. This process produces brown rice and byproduct husk, used as fuel, bedding, or organic material. Brown rice. Brown rice still has the bran layer and embryo intact, making it more nutritious but with a shorter shelf life. Polishing. The bran layer and embryo are removed by milling/polishing to produce white rice. Byproducts of polishing are bran and embryo, which are used for animal feed, rice bran oil extraction, or as nutritional supplements. White rice. The final polished product, commonly consumed, with a longer shelf life but lower fiber and micronutrient content compared to brown rice.
1. Parboiling. Parboiling is a hydrothermal process applied to rough, unmilled, rice before milling. It involves 3 main steps. Soaking the paddy in water to allow moisture penetration. Steaming to gelatinize the starch and harden the kernels. Re-drying to a safe moisture content for milling. Parboiling improves milling recovery, reduces broken grains, enhances nutritional retention, and increases the storage life of rice. 2. Curing. Freshly harvested rice, new rice, has a lower swelling capacity and tends to produce a thick, viscous gruel during cooking. Curing involves storing milled rice for a specific period under controlled conditions to improve cooking quality, aroma, and texture. Proper curing reduces stickiness and enhances consumer acceptability. 3. Milling. Milling is the mechanical process of removing the inedible outer husk and bran layers from rough rice to produce edible rice. It consists of dehusking, removing the outer husk to obtain brown rice. Bran removal. Milling further to produce white rice. Efficient milling ensures maximum head rice recovery with minimal breakage. 4. Polishing, whitening. Polishing is the final step in milling, involving the removal of any remaining fine bran particles from the surface of the rice kernel. This process, often done 2 to 3 times, gives the rice a smooth, bright, and appealing appearance. While polishing improves appearance and shelf life, it can also reduce nutrient content by removing trace amounts of vitamins and minerals present in the bran layer.
System of Rice Intensification, SRI, in paddy cultivation. The System of Rice Intensification, SRI, is an innovative method of paddy cultivation that was first developed in Madagascar in the 1980s. Since then, it has been adopted in more than 25 countries worldwide due to its potential to enhance yields, reduce input costs, and promote sustainable water and soil management. Key principles and practices. Seed rate. A very low seed rate of 5 kg per ha is used compared to conventional practices, reducing seed costs and minimizing plant competition. Spacing. Wider spacing of 25 by 25 cm between hills allows for better root and canopy development, improved aeration, and reduced competition for nutrients, water, and sunlight. Seedling age and transplanting. Only single seedlings, 10 to 12 days old, are transplanted to avoid root damage and to promote vigorous tillering. Young seedlings establish quickly and produce more productive tillers than older seedlings. Nutrient management. Recommended fertilizer application: 120 kg nitrogen, 60 kg P2O5 and 40 kg K2O per hectare. Organic manures or compost can also be incorporated to improve soil structure and microbial activity. Water management, not in your original text but essential for SRI. Unlike traditional flooded systems, SRI uses intermittent irrigation, keeping the soil moist but not continuously submerged. This encourages deeper root growth, improves soil aeration, and reduces methane emissions. Advantages of SRI. Higher yields with lower seed requirement. Better root development and nutrient uptake. Reduced water use compared to conventional methods. Improved grain quality and higher market value.
Scented rice. Scented rice refers to special aromatic varieties valued for their fragrance, flavor, and superior grain quality. These varieties command a premium price in domestic and international markets due to their unique cooking and eating qualities. 1. Popular varieties. Pusa Basmati Series, Pusa Basmati 1, Pusa Basmati 14, Pusa Basmati 370. Pusa Sugandha Series, Pusa Sugandha 2, Pusa Sugandha 3, Pusa Sugandha 5. Traditional Basmati varieties, Basmati 370, Kasturi Basmati. State-specific aromatic varieties. Bihar, Rajendra Suhasini, Rajendra Bhagwati, Rajendra Kasturi, Bhagalpur Katani. Haryana, Hisar Basmati. 2. Nutrient management. Recommended dose: 100 kg nitrogen, 60 kg P2O5 and 60 kg K2O per hectare. Balanced nutrition is critical for optimum yield, maintaining grain aroma, and ensuring high milling recovery. 3. Harvesting. Harvest at the physiological maturity stage to preserve the natural aroma and maximize head rice recovery around 65%. Delayed harvesting may lead to aroma loss, grain shattering, and reduced quality. 4. Yield. Average productivity ranges from 35 to 40 quintals per ha, depending on variety, agronomic practices, and environmental conditions. 5. Special considerations. Gentle handling during harvesting, threshing, and milling is essential to prevent breakage and aroma loss. Proper curing and storage under low moisture and cool conditions help maintain fragrance for longer periods.
Semi-deep water rice. Semi-deep water rice varieties are adapted to environments where fields remain submerged under 25 to 50 centimeters of water for extended periods during the growing season. These varieties possess tolerance to submergence and are suitable for flood-prone regions. Popular varieties: Mojuka, Jalmagna, Charkia 59, Jaladi, Vaidhai, Janaki, Sudho, Amulya, Nalini, Rajendra Masuri. Nutrient requirement: 60 to 80 kg nitrogen, 30 kg P2O5 and 30 kg K2O per hectare depending on soil fertility and water depth. Yield potential: 30 to 35 quintals per hectare under proper management. Special trait: ability to survive and produce economic yield in partially flooded conditions where normal varieties fail. Hybrid rice. Hybrid rice technology exploits heterosis, hybrid vigor, to achieve significantly higher yields compared to conventional varieties. It has been widely adopted in several rice-growing states of India. Seed rate: 5 to 6 kg per hectare, due to vigorous growth and wider spacing. Popular hybrids: PHB 71, KRH2, PA 644, Pusa RH10, Scented Hybrid, Pant Hybrid Rice 1. Nutrient requirement: 120 kg nitrogen, 60 kg P2O5 and 30 kg K2O per hectare for optimum productivity. Yield potential: 100 to 110 quintals per hectare under recommended management practices. Advantages: higher yield, better input use efficiency, and suitability for both irrigated and rain-fed ecosystems, depending on hybrid type. Saline-tolerant rice. Saline and alkaline soils restrict the productivity of most rice varieties due to poor germination, stunted growth, and reduced grain filling. Specially bred varieties tolerate high salt concentrations and perform reasonably well in such conditions. Popular varieties: Narendra Usar 1, Narendra Usar 3, CSR 5, CSR 10, CSR 23. Special trait: tolerance to salinity and acidicity during critical growth stages. Cultivation tip: apply gypsum or other soil amendments along with balanced fertilizers to enhance productivity under saline alkali conditions. Direct seeded rice, DSR, cultivation. Direct seeded rice is a method of crop establishment in which seeds are sown directly into the field, without raising and transplanting seedlings from a nursery. This method is particularly advantageous under certain conditions and has gained attention as a labor and water-saving technology. Key features and advantages. Suitability. Highly beneficial during the dry season, when water supply can be easily regulated. Recommended in areas with labor shortages, especially during the peak transplanting period. Most suitable where weed infestation is not severe, as weed control in DSR can be more challenging than in transplanted rice. Early maturity. The crop matures 7 to 10 days earlier than the transplanted crop, allowing timely harvest and facilitating sequential cropping. Cost saving. Eliminates the need for seedbed preparation, nursery management, and transplanting operations. Reduces expenditure on plant protection associated with transplant shock and handling. Overall cost of production is reduced compared to the transplanting method. Timely sowing. Enables quicker crop establishment, ensuring sowing is done at the optimal time for higher yield potential. Limitations. Weed management is critical and may require integrated weed control measures. Germination and crop stand depend heavily on seed quality, soil moisture, and precise land preparation.
Common diseases of rice and their management. 1. Seedling blight. Causal organism, Sclerotium rolfsii. Favorable conditions. Warm and wet soil conditions promote the development of the disease. Symptoms. Dark-colored rot at the base of seedlings. White, cottony mold growth on the lower parts of plants. In heavily infested soils, large numbers of seedlings may die, often in distinct rows. Management. Use well-drained nursery beds. Treat seeds with recommended fungicides before sowing. Avoid excessive irrigation in nurseries. Remove and destroy infected seedlings. 2. Rice blast. Causal organism, Pericularia grisea, sin. Magnaporthorizii. Symptoms. Rice blast can infect all above-ground parts of the plant. Based on infection sites, it is classified as leaf blast, spindle-shaped lesions with gray centers and brown margins. Collar blast, lesions at the junction of leaf blade and sheath, causing leaf death. Node blast. Infection at the node, leading to blackening and breaking of the stem. Neck blast, rotting and breaking of the panicle neck, resulting in poor grain filling. Management. Grow resistant varieties suitable for the local region. Avoid excessive nitrogen application, especially in a single dose. Maintain proper plant spacing for good air circulation. Spray recommended fungicides, e.g., tricyclazole or isoprothiolane, at early symptom appearance.
3. Brown spot, Helminthosporiose leaf spot. Causal organism, Helminthosporium oryzae, sin. Bipolaris oryzae. Symptoms. Affects leaves, glooms, coleoptils, leaf sheaths, and panicle branches. On leaves and sheaths, symptoms appear as oval or circular brown spots with distinct margins. Spots are evenly scattered across the leaf surface, often coalescing under severe infection. Management. Use disease-free seeds and resistant varieties. Follow proper crop rotation to avoid pathogen buildup. Apply balanced fertilizers, particularly adequate potassium, to strengthen plants. Spray recommended fungicides, e.g., propiconazole or mancozeb, when symptoms first appear. 4. False smut, green smut. Causal organism, Eustulaginoidea virens. Symptoms. Occurs after flowering, affecting individual grains in the panicle. Infected grains are transformed into greenish, velvety spore balls that enlarge over time. Smut balls may later turn yellowish or orange as they mature. Management. Use healthy, certified seed. Avoid excessive nitrogen application during heading stage. Synchronize planting to minimize extended flowering periods. Spray fungicides such as propiconazole at booting stage and repeat if necessary. Sheath blight. Causal organism, Rhizoctonia solani. Symptoms. Initially appears as ellipsoid or oval spots on the leaf sheath, sometimes irregular in shape. Spots are greenish-gray at first, later turning greenish-white in the center with distinct brown margins. Lesions may enlarge and merge, causing rotting and lodging in severe cases. Management. Avoid dense planting to improve air circulation. Apply balanced fertilizers, avoiding excess nitrogen. Remove infected plant debris from the field after harvest. Spray recommended fungicides, e.g., validomycin or carbendazim, at early symptom appearance.
Seed selection and treatment for disease management in rice. 1. Selection of healthy seeds. Always select mature, healthy, and disease-free seeds. For blast prevention, treat seeds with tricyclazole 75 wettable powder at 0.6 gram per kilogram seed. 2. Seed treatment before storage. Before storing, seeds should be cleaned, thoroughly dried, and treated with one of the following fungicides. Captan 50 wettable powder at 3.0 gram per kilogram seed. Mancozeb 75 wettable powder at 3.0 gram per kilogram seed. Mancozeb 75 wettable powder at 2.0 gram per kilogram seed plus carbendazim 50 wettable powder at 1.0 gram per kilogram seed. 3. Pre-sowing seed treatment. To minimize seed-borne disease transmission, soak seeds overnight in one of the following solutions. Tricyclazole 75 wettable powder at 60 gram per 100 liters water. Edifenfos 50 emulsifiable concentrate C at 100 milliliters gram per 100 liters water. Important, do not treat sprouted seeds with fungicides, as this can damage the plumule or radicle. Sprays of any of the following fungicides are recommended to control the disease in paddy. Rice stem borer. Scientific name, Serpophaga incirculars, yellow stem borer. Nature of damage. Larvae bore into the stem and feed on the internal tissues. At vegetative stage, causes dead hearts, central shoot dries up and can be easily pulled out. At reproductive stage, causes white ears, panicles emerge but grains remain chaffy. Management. Clip seedling tips before transplanting to remove egg masses. Encourage natural enemies like Trichogramma japonicum. Avoid excessive nitrogen application. Apply recommended insecticides, e.g., Cartap hydrochloride, fipronil, at the right stage. Brown plant hopper, BPH. Scientific name, Niloparvata lugens. Nature of damage. Suck sap from the base of tillers, leading to hopper burn. Damaged plants turn brown and dry quickly, often in patches. Also acts as a vector for grassy stunt and ragged stunt viruses. Management. Maintain proper plant spacing to reduce humidity in the crop canopy. Avoid indiscriminate use of broad-spectrum insecticides that kill natural predators. Apply selective insecticides, e.g., buprofezin, pymetrazine, when BPH exceeds ETL, economic threshold level.
Paddy grasshopper, Oxyanatigilla or Hieroglyphus banyan. Rice hispa, Diclodispa armigera. Management practices. Mechanical control. Remove and destroy leaves infested with rice hispa to prevent further spread. Scrap and clean field bunds to remove grasses and weeds, thereby destroying pest eggs and reducing future infestations. After harvesting, plow the field to expose and destroy pest eggs lying in the soil. Chemical control. For early infestation, spray endosulfan 35 emulsifiable concentrates at the rate of 140 ml per 100 liters of water, dimethoate 30 emulsifiable concentrates at the rate of 100 ml per 100 liters of water, or fozolone 35 emulsifiable concentrates at the rate 140 ml per 100 liters of water. For grasshopper control, apply carbaryl 10% wettable powder dust on the crop and grassy bunds at the rate 15 to 20 kg per ha, preferably in the morning hours, 15 days after transplanting. If pest population reaches the economic threshold level, ETL, repeat the treatment as necessary. For long-lasting protection, spray a residual insecticide such as chlorpyrifos 20 emulsifiable concentrates at the rate 100 ml per 100 liters of water or endosulfan 35 emulsifiable concentrates at the rate 140 ml per 100 liters of water.
Summary. Scientific practices for optimizing paddy yield involve an integrated approach that includes selecting high-yielding and disease-resistant rice varieties, adopting precise water management techniques like alternate wetting and drying, AWD, and applying balanced fertilization based on soil testing. Timely sowing, use of certified seeds, and effective pest and weed management also play a critical role in boosting productivity. Incorporating mechanization, such as using seed drills and harvesters, enhances efficiency, while modern practices like System of Rice Intensification, SRI, further improve yield potential. Overall, combining traditional knowledge with advanced agricultural technologies ensures sustainable and optimized paddy production.